These are my homemade DNA and RNA models. On the left is DNA and on the right is RNA. To make my models, I used a bent paperclip to represent the strong phosphate sugar backbone, and I used marshmallows that I wrote on with blue pen to represent the various nitrogenous bases. At first look, it can be determined that there are various similarities and differences between DNA and RNA molecules. DNA specifically is a double helix. It has two strands that wrap around to create the double helix structure. The nitrogenous bases of DNA are adenine, thymine, guanine, and cytosine. These will always be the nitrogenous bases for DNA. Because of complementary base pairings, adenine will always pair with thymine and guanine will always pair with cytosine. However, RNA is a single-stranded molecule. RNA has very similar nitrogenous bases, however, there is one major key difference. RNA also has adenine, guanine, and cytosine. However, instead of thymine, RNA has uracil. In DNA, hydrogen bonds hold the nitrogenous bases together, while covalent bonds hold the phosphate sugars together to form the backbone. Covalent bonds also hold the phosphate sugars together on RNA to form the strong backbone as well. For my models, I could not go into enough detail in the phosphate sugar backbones to display this. However, I'm still going to explain it as it is a key difference between the two molecules. The two DNA and RNA have different pentose sugars. RNA has one more oxygen than DNA, and DNA has one less oxygen than RNA. This is important to remember when drawing the diagram in major detail, as IB requires. Lastly, it is important to remember that DNA is a double-stranded helix, and the two strands are anti-parallel, meaning they go in opposite directions.